I will make two videos to cover this particular competency, that is the metabolic processes that take place in specific organs in the body in the fed and fasting states. Part 1 will cover the fed state, while in part 2 I'll be describing the fasting state. I will begin with the overview of the feed fasting cycle. We begin by looking at this graph, and we will see how we maintain blood glucose level with time. In this graph, time 0 is having consumed the last meal and assuming that meal has some carbohydrate. We first maintain our blood glucose level from the carbohydrate in that meal, that is the exogenous carbohydrate. Blood glucose rises, that causes an increase in blood insulin, and we utilize the blood glucose within 3 to 4 hours. Usually after couple of hours after the meal, blood glucose level drops to below 100 milligram per deciliter, and insulin level drop down close to their basal level, and there is simultaneous rise in the level of the hormone glucagon, and liver glycogen is activated to be broken down to release glucose in the blood to maintain blood glucose level. So during time period between meals, it is mostly liver glycogen that maintain our blood glucose. If we don't eat for about 4 hours to overnight, the process of gluconeogenesis begins. That is, synthesizing glucose by the liver and by the kidney cortex from non-carbohydrate precursors such as cholesterol, lactate, and amino acids, as well as lipolysis, so releasing fatty acid from adipose stores. At this stage, most tissues in the body switches over to oxidizing fatty acids, thus sparing glucose for utilization by the brain and the red blood cells. By the day of fasting, that is 24-hour, essentially no glycogen is left in the liver, and we become totally dependent on gluconeogenesis to supply blood glucose. If the fast continues 1 to 2 days, then the process of ketogenesis with the synthesis of ketone bodies are activated in the liver. Ketone bodies are made from acetyl coenzyme A, which comes from fatty acid oxidation occurring in the liver. These ketone bodies supply energy particularly to brain but also to heart muscles and kidneys. This helps spare glucose. So gluconeogenesis levels begins to drop and maintain at the constant low levels throughout the length of the fast, and this helps spare proteins, since muscle proteins are the main suppliers of substrate for gluconeogenesis. We all eat periodically throughout the day. However, our organs, especially the brain, need a continuous supply of glucose. How does the body meet this constant demand for energy? The body processes food we eat both to use immediately and, importantly, to store as energy for later demands. If there were no method in place to store excess energy, we would need to eat constantly in order to meet energy demands. Distinct changes in enzyme activities occur in the absorptive and the fasting states that facilitates energy storage. Energy storage occur during absorptive state and to make stored energy available during times of fasting and starvation. First, I'll explain how the flow of intermediates through metabolic pathways are controlled during absorptive state. During absorptive state, the flow of intermediate through metabolic pathways are controlled by the availability of substrates, allosteric regulation of enzymes, covalent modification of enzymes, induction and repression of enzyme synthesis. Allosteric changes mostly affect rate limiting reactions. Consumption of meal stimulates glycolysis in the liver. How it regulates? It does so by increasing fructose 2,6 bisphosphate, which is an allosteric activator of phosphofructokinase 1. Since glycolysis has already been covered, you must be aware of this enzyme. However, gluconeogenesis is inhibited at the same time by fructose 2,6 bisphosphate, an allosteric inhibitor of fructose 1,6 bisphosphatase. Activity of many enzymes is regulated by the addition or removal of phosphate groups to serine, threonine, or tyrosine residues of the protein. In absorptive state, most of the covalently regulated enzymes are active in the dephosphorylated form, which is shown in green. However, there are 3 exceptions in which the enzymes are active in their phosphorylated form, or you can say they are inactive in their dephosphorylated form, and it include glycogen phosphorylase kinase, glycogen phosphorylase, and hormone sensitive lipase. Increased or decreased enzyme synthesis causes changes in the number of enzyme molecules. These are enzymes that are needed under specific physiologic condition, and these enzymes are subjected to regulation of synthesis. Fed state causes the activation causes the elevation of enzyme that results in increased synthesis of enzymes such as the enzyme of fatty acid synthesis, that is acetyl coenzyme A carboxylase fatty acid synthase. In contrast, in fed state, elevated glucagon induces expression of enzymes such as phosphoenol pyruvate carboxykinase. The absorptive state or the fed state occurs after a meal when the body is digesting the food and absorbing the nutrient. This is a state when one has plenty of food in the intestine and body is digesting it and absorbing it. Purpose of the absorptive state is to store energy so that it can be used later. Typically, the absorptive state is 2 to 4 hour period after ingestion of a normal meal. So during absorptive state, there is transient increase in the plasma levels of glucose, amino acids, and triacyl glycerol. Liver is a central player. Drainage from gut and pancreas travel via the hepatic portal vein to liver. Liver is basically ideally placed to regulate fuel passage elsewhere. After a meal, the liver is bathed in a blood which has absorbed nutrients and elevated levels of insulin, which is secreted by the pancreas. Liver takes up carbohydrates, lipids, and most amino acid. These nutrients are then metabolized, stored, or routed to other tissues. Liver is basically preventing potentially broad fluctuations in the availability of nutrients for the peripheral tissue. Now I'll explain the metabolic profile of liver in fed state. We will begin the discussion with the carbohydrate metabolism in liver. Liver is normally a glucose producing rather than a glucose utilizing tissue. But after a meal containing carbohydrate, the liver becomes a net consumer, retaining approximately 60 gram of every 100 gram of glucose presented by the portal system. This increased use reflects increased glucose uptake by the hepatocytes. Liver take up glucose through its insulin independent glucose transporter, that is GLUT2. GLUT2 has low affinity and a high Km for glucose and takes up glucose only when the blood has high level of glucose. Hepatic glucose metabolism is increased due to increased phosphorylation of glucose. When there is elevated levels of glucose within the hepatocyte as a result of elevated extracellular levels, the enzyme glucokinase phosphorylates glucose to glucose 6-phosphate. Glucokinase comes into play because it has a high Km for glucose, and glucokinase is not inhibited by the product, that is glucose 6-phosphate, in contrast to hexokinase. Hepatic glucose metabolism is increased also because of increased glycogenesis, that is the synthesis of glycogen from glucose 6-phosphate, by the activation of enzyme glycogen synthase. Both it is activated both by dephosphorylation and because of the increased availability of glucose 6-phosphate, its allosteric effector. Pentose phosphate pathway is activated by the increased availability of glucose 6-phosphate and also because of the active use of NADPH in hepatic lipogenesis. Only during the absorptive state or absorptive period, glycolysis is significant in the liver following a carbohydrate-rich meal. Elevated insulin to glucagon ratio stimulates the conversion of glucose to pyruvate, and this is due to increased amount of the regulated enzymes of glycolysis such as hexokinase, phosphofructokinase 1 and pyruvate kinase. I am not going into the details of how the activities of these enzymes are regulated, as this has already been covered. The acetyl coenzyme A is either is used as a substrate for fatty acid synthesis, or it is oxidized for energy by the TCA cycle. Hepatic glucose metabolism is also increased due to decreased production of glucose. Glycolysis and glycogenesis, the pathway that promotes glucose storage, are stimulated in the liver in the absorptive state. Gluconeogenesis and glycogenolysis, the pathways that generate glucose, are inhibited or decreased. Pyruvate carboxylase, which catalyzes the first step in gluconeogenesis, is largely inactive due to low levels of acetyl coenzyme A. Its allosteric activator, acetyl coenzyme A, is used for fatty acid synthesis. The high insulin to glucagon ratio also favors inactivation of other glucogenic enzymes such as fructose 1,6-bisphosphatase. Glycogenolysis is inhibited by dephosphorylation of glycogen phosphorylase and glycogen phosphorylase kinase. Now I'll explain how the fat is metabolized in the liver in fed state. The increased synthesis of fatty acid during fed or the absorptive state. Liver is primary tissue where de novo synthesis of fatty acid occur. Glucose can be converted to triacylglycerol, but for this it requires glycerol and fatty acid. In order to convert glucose to fatty acid, glucose is converted to pyruvate and acetyl coenzyme A. Fatty acid synthesis, which occur in the absorptive period, is because of the availability of the substrate acetyl coenzyme A from glucose and oxidative degradation of certain amino acids, and NADPH from glucose metabolism, and by the activation of acetyl coenzyme A carboxylase. You must be aware of what acetyl coenzyme A carboxylase does. During the fed state, triacylglycerol synthesis is favored because fatty acyl coenzyme A are available both from de novo synthesis from acetyl coenzyme A and from hydrolysis of triacylglycerol component of chylomicron remnants removed from the blood by the hepatocytes. Glycerol 3-phosphate, the backbone for TAG synthesis, is provided by glycolysis. The liver packages triacylglycerol into very low density lipoprotein or VLDL particles that are secreted into the blood for use by extrahepatic tissues, particularly adipose and muscle tissues. Now we will see how the amino acids are metabolized in the liver in fed state. In the absorptive state, we have a large amount of amino acids present in the liver, then it can be used for the synthesis of protein and other nitrogen containing molecules. The surplus amino acids are not stored but are either released into the blood or for other tissues to use in protein synthesis, or it can be the amino acids can undergo deamination, or it can be deaminated, with the resulting carbon skeleton being degraded by the liver to pyruvate acetyl coenzyme A or TCA cycle intermediates. These metabolites can be oxidized for energy or used in fatty acid synthesis. The liver has a limited capacity to degrade branched chain amino acids such as leucine, isoleucine, and valine. They pass through the liver essentially unchanged and are preferentially metabolized in the muscle. The body does not store protein in the same way that it maintains glycogen or TGA reserve or TAG reserves. However, a transient increase in protein synthesis of hepatic proteins does occur in the absorptive state, resulting in replacement of any proteins that may have been degraded during the previous post-absorptive period. Now we will discuss the metabolic profile of adipose tissue in fed state. See, adipose tissue is second only to liver in its ability to distribute fuel molecules. In a 70 kg man, white adipose tissues weighs approximately 14 kg or about half as much as the total muscle mass. Nearly the entire volume of each adipocyte in white adipose tissue can be occupied by a droplet of triacylglycerol. Now we will discuss the carbohydrate metabolism in adipocytes. Carbohydrate metabolism in adipocytes is increased due to increased glucose transport. Glucose is taken up via insulin-sensitive GLUT4 recruited to the cell surface from the intercellular vesicles. The glucose is phosphorylated in adipocytes by the enzyme hexokinase. The increased intracellular availability of glucose result in enhanced rate of glycolysis. In adipose tissue, insulin directs adipocytes to take up fatty acids and glycerol for triacylglycerol synthesis, that is lipogenesis. Glycolysis in adipose tissue is used. The glycolysis in adipose tissue is used for supplying glycerol 3-phosphate for triacylglycerol synthesis, as adipose tissue lacks glycerol kinase. Adipose tissue can metabolize glucose by means of pentose phosphate pathway, or which is also called as HMP shunt pathway, thereby producing NADPH, which is essential for fat synthesis. However, in humans, the de novo synthesis is not a major source of fatty acid in adipose tissue except when refeeding a previously fasted individual. In fed state, elevated levels of glucose and insulin favor storage of triacylglycerol. Elevated insulin inhibit hormone-sensitive lipase, thereby inhibiting lipolysis in fed state. Now we will discuss the metabolic profile of resting skeletal muscles in fed state. Muscle take up glucose via GLUT4 transporter in fed state and use it for energy and glycogen synthesis, and takes up amino acid including branched chain amino acid for protein synthesis. Now we will see how the carbohydrate is metabolized in the muscles during fed state. Increased glucose transport. The rise in the plasma glucose and insulin after carbohydrate rich meal lead to an increase in glucose transport into the muscle via GLUT4, thus reduces blood glucose. In muscles, glucose is phosphorylated to glucose 6-phosphate by hexokinase and metabolized to provide energy needs of the cell. The increased insulin to glucagon ratio and the availability of glucose 6-phosphate favors glycogen synthesis. Action of lipoprotein lipase releases fatty acids from chylomicrons and VLDL, but in fed state, glucose is the primary source of energy for muscles. Now we'll see how the amino acids are metabolized in the muscles during fed state. In the absorptive period, after ingestion of a meal containing protein, there is enhanced uptake of amino acid by muscles and it is used for protein synthesis. So there is enhanced uptake of amino acid and this amino acid is used for the synthesis of protein. This helps replace protein degraded during the post-absorptive period. Muscle is the principal site for the degradation of branched chain amino acid because it contains the required transaminases. The branched chain amino acids, they are taken up by the muscles while they escape metabolism by the liver and they are used for the protein synthesis. Now we'll see how the brain behaves in fed state. The brain is vital to the proper functioning of all organs of the body. Special priority is given to its fuel need. In the fed state, brain exclusively uses glucose through its glucose transporter, that is GLUT1, which is insulin independent, and it completely oxidizes glucose to carbon dioxide and water. The brain has no significant stores of glycogen, and because of which brain is completely dependent on the availability of blood glucose. If blood glucose levels fall below 40 milligram per deciliter, the normal fasted blood glucose is between 70 to 100 milligram per deciliter. So if it falls below 40 milligram per deciliter, then the cerebral function will be impaired. The brain also lacks significant stores of triacylglycerol, and the fatty acids circulating in the blood make little contribution to energy production because fatty acids bound to albumin do not efficiently cross the blood-brain barrier. This is the overall picture of the various organs after the meal. Together, this presents the picture as insulin are stimulating growth and building storage molecules throughout the body.